So what I'd like to do today is give you a couple of vignettes of how an ocean scientist will use synchrotron X-ray techniques to understand things that we care about. So in this particular case, it's iron and manganese cycling in the ocean. So I'll give you a little bit of an intro to why you should care about iron and manganese cycling in the ocean, and give you two case studies for an iron one, which is actually quite old now. It's over 10 years old, but it sort of set the stage for how I think about understanding these kinds of things in the ocean. And then a much more recent one, which is a manganese cycling in the ocean. Whoops. Okay, so why study iron? So biological productivity in about 30% of the ocean is limited by iron. So here's a map showing in background that shaded color is the concentration of basically major nutrients. In this case, it's nitrate divided by 16. I won't go into why, it doesn't matter. But the light blue color is very, very low major nutrients. The green colors are higher major nutrients. And then the red dots are showing you different experiments over the past decades that try to test what nutrient is the main controlling factor for biological productivity. And anywhere you see a red dot is where it was confirmed that iron is in fact the limiting nutrient. So all of these red dots you'll see end up appearing in these places where you have high major nutrients. And the idea is that a major nutrient, nitrate, phosphate, you need that for proteins, lipids, DNA. But iron is an essential micronutrient. You need it for many biochemical functions. And in these particular places, even though you have plenty of major nutrients, you don't have enough micronutrient iron. So we really need to understand iron based on these experiments. And you can go back in time as well. And what this is showing you is a sediment core. The sediment core was actually taken from around here. So in what we can see from modern studies is in this iron limited area. And this is going back in time from the present here to 1 million years ago. And I'm showing you the reconstruction of PCO2, so atmospheric CO2 concentrations, iron deposition in the sediment core, and biomarkers that indicate productivity. And without going through the details, what I just want you to notice is that when PCO2 is low, so when atmospheric CO2 is low, iron is high, productivity is high. So there's, and this feature of seesawing up and down PCO2, so that's our glacial, interglacial cycles, warm interglacial, cold glacials. In the cold glacials, a lot of iron, more productivity. This is a repeating pattern. We know it's happened in the past. There's this very strong correlation. And of course, correlation isn't causation, but there's a lot of reason to believe that there is a causal effect. There are feedbacks, but there is a causal effect. Iron causes more productivity. Productivity draws down CO2 in the atmosphere. So we need to care about iron. So as a result, all climate models now have iron in their models because iron is such an important control on productivity for so much of the ocean that this has a global effect. And climate model, so what this is showing you was a relatively recent intercomparison of iron in climate models, of how iron is modeled in climate models. And here there's a handful, say a dozen, of different global climate models. All of these sort of, not all of them, many of them feed into the main climate models that are being used to predict future climate. On the y-axis is the mean or average iron concentration in the ocean. So can the model reproduce iron distributions? Not too bad, a factor of 2 variation between the models. But if you now look at other metrics that assess how well the models can do iron. And what this is looking at is the fluxes of iron estimated by these different models, these global fluxes as a result of the sum of all of these sources. And I'll show you a diagram that sort of talks about what the sources are. But here are the main sources. If you add them all up, you now see that the y-axis is a log scale, and the different models vary not by a factor of 2, but 2 orders of magnitude. So the models are relatively good at reproducing the distribution, but horrible agreement on the magnitude of the fluxes. So what this is telling us is we can come up with reasons for why iron is high in one place and another is low at another place, but our reasons do not agree with each other. And we need to better constrain the sources and the sinks, what's going on with the fluxes that are coming in and what's going on with the fluxes going out. And that is part of what motivates what I do. So I'll move from iron to manganese. So manganese is also a micronutrient. It's not nearly as important of a micronutrient in that it is never or rarely the limiting nutrient. It doesn't control productivity. It's absolutely necessary, but it's not the thing that is keeping growth. The reason I'm interested in manganese is that in its particulate form as an oxide, it is an incredibly efficient scavenger. It has manganese oxide has extremely high specific surface area, and as a result, it is very efficient at scavenging other trace metals, including cobalt, molybdenum, copper, nickel, all sorts of trace metals that are of economic interest. And this is why manganese nodules and ferromanganese crusts are at the moment being targeted for deep sea mining exploration. And I did a Google search last night, and these are just a couple of articles in the news that I saw pop up, and this is just in the last few weeks. So this is gaining a lot of momentum. There's a lot of companies that are currently exploring the potential for manganese nodules and crusts for minerals that for metals that we need for batteries for our future electric lifestyle. So my interest is much removed from that. I'm interested in how can we understand this sort of scavenging process, that part that allows us to maybe get to mining, whether you agree with that or not. What are the sort of what's going on with the chemistry, the geochemistry of manganese that allows it to scavenge other trace metals? So I lump these two together because they have similarities in their overall sort of geochemical cycling. In the dissolve phase, both are essential micronutrients. Both of them have active redox cycles, iron cycles between iron 2 and iron 3 in the ocean, manganese cycles between manganese 2, 3, and 4. Both of them are more soluble in their reduced form, so iron 2 and manganese 2, and therefore more bioavailable. Since both of them are nutrients, then if they're in that soluble form, they will be more bioavailable. And in their oxidized forms, both of them are very insoluble. So once they get oxidized to iron 3 or to manganese 4, they precipitate, and typically as some sort of oxide or oxyhydroxide. And then both of them do form oxides with extremely high specific surface area. So they're both very efficient scavengers. I'm not focusing on iron as a scavenger in this talk, but it is also a scavenger. Its oxidation is just, well, that's not true. Never mind. I'll focus on manganese. Okay, so here is a sort of general schematic of where the different parts of the Earth system, what the sources of iron and manganese are to the ocean from different parts of the Earth system. So here's my schematic of the ocean. We have aerosol mineral dust deposition that will bring, so basically iron and manganese are relatively high concentration, actually quite high concentration in Earth's crust. So the fact that it's low in the ocean is because even though the Earth has plenty iron and manganese, they're very insoluble. And so you need to be close to a source, to some sort of terrestrial source in order to get it. And you also have to have a mechanism to render it soluble. So you can get atmospheric dust deposition, you can have river introduction. I boxed these and color coded them by whether it's bringing in more of the oxidized form or more of the reduced form. It doesn't really matter. But you can have groundwater introduction that tends to bring in reduced forms. The altered oceanic crust tends to have reduced iron and manganese. Hydrothermal fluids will also bring in reduced iron and manganese. Shelf pore fluids from sediments will also bring in reduced iron and manganese. And then you could have redox cycles. This particular diagram was made for iron, but there is also a redox cycling of manganese. So they have these broad similarities, and we can think of them similarly. But if you then look at how they're distributed, they're very different. So I'll just walk you through how you would read this. Here is on the x-axis is latitude. So we're going from 60 north up here to 60 south down here. So we're doing this transect down the Atlantic. On the y-axis is depth. And so what we're doing is a slice down the Atlantic. And we're looking now at the colors tell you the concentrations. And up here we have the concentrations of dissolved iron. So yellow is high, blue is low, and because it's a limiting nutrient, it's going to be low in the surface, and you see that it's this dark blue, this low concentration most places in the surface, and generally higher at depth, but it's quite splotchy, as you can see. And then manganese, again, same way you read it, this is a slice, yellow is high, so you see that manganese is really high in the surface. So I told you it was a micronutrient, but it's never limiting, it's super high in the surface, and then it gets low in the deep. So despite their similarities, they are redox active, they have similar sources, they have redox cycling, they are both micronutrients, they have vastly different distributions in the end. So that means that there are differences in their chemistries that we need to take into account, and we need to model differently if we're to understand what controls their distributions. Okay, so I focus, I would say that most people in oceanography of my ilk, chemical oceanographers, care about the dissolved phase because that's the bioavailable phase. But I've been focusing on the particulate phase because that holds a lot of information. So the mineralogy and the redox speciation of the particulate phases can contain information about the sources and the cycling, their sort of chemical history, their geochemical history, by just looking at their mineralogy and speciation. And of course, as everyone who is watching this will know, X-ray absorption techniques are great for this, for probing mineralogy, redox speciation of elements without too much without doing too much to the sample. There are challenges, though, to working with marine samples, and that's what I've spent most of my career sort of trying to overcome and trying to figure out how to still get good data and get information that is useful to an oceanographer working within our limitations. So the main limitation is that open ocean water column samples have very low concentrations. I've worked tens to hundreds of nanogram per centimeter squared. I'll show you later that we collect samples onto a filter and then just put the filter in front of the beamline. Very low, sometimes it's actually more like single nanograms to tens, more like. And that means that excess doesn't really work unless you have very, very high concentrations. So we can't really rely on excess to understand what's going on generally with our samples, because in general, the concentrations are too low. There are exceptions, and I'll show you a couple. Beam damage is a problem. So just because we have low concentrations, we can't get around that by just increasing flux, because then we end up inflicting beam damage. So we end up having to do things very quickly, quick XAS to figure out whether we are causing beam damage. We have to do things in cryo environments. We have to try to keep things very cold. This is especially the case for manganese, we found. And then the third thing is, as I'll show you, there's a lot of heterogeneity in the speciation and mineralogy of iron and manganese. So even though you might be tempted to want to understand a lot of the detail of the fine scale variation, if you of any particular sample, you're going to have a very hard time trying to understand the broad scale of what's going on if you don't pull back. And there's such high heterogeneity that you really need to focus on high throughput strategies in order to get the sort of broad overview of what's going on. And then you can go and focus and learn everything you want to your heart's content about one specific sample if you want, if you have enough concentration to do so. Oh, and this is my plug for Sarah, who's on the line. She led this paper that was just published 2 days ago on a workshop that was a year ago today, that a few people, including myself, spoke about how one uses synchrotron probes to look at marine samples. And so I'm just giving you my view and just a couple of case studies from what I do in my lab. And if you're interested, you can go to this paper. This paper links to video recordings of this workshop, and you can find out what lots of other people are doing as well. So plug for that. Okay, so what do our samples look like? I alluded very briefly to the fact that we have samples on a filter. So imagine seawater with suspended particles in it. We put this pump down into the seawater, and we pump water. I think I have I do. Let me just go to that. This pump is hung on a wire and then it's battery operated. There's a motor and it pulls water, seawater through these filter holders. And we pump for about 4 hours. We get about 1,500 liters of seawater through these filters, through these 2 flow paths. We have 2 flow paths, so we could have 2 different filter types because these are 142 millimeter filters, which we can then divide, cut up into slices or punch holes, and then distribute to many different PIs or just to many different people within my group to measure different things. The idea is if we have a very even distribution of particles, we can make lots and lots of measurements and put them together and learn a lot of things. So we need 2 different filter types in order to be compatible with all the measurements that we're trying to make. We have a large size fraction. So particles in the ocean have different sizes. Most of the sediments and the plankton are what we call put into the small size fraction, say 1-ish to 50 micron-ish. These are what we call suspended particles. And then these particles can aggregate to large particles. These are responsible for the flux, the sinking, and the removal of particles from the ocean. So we also collect large particles. After you filter all of these particles, you have this sort of mishmash of different types of particles on your filter. So here's my little cartoon of the filter. I've colored it green because green is meant to show biogenic particles. So particulate organic matter from photosynthesis that dominates, especially in the upper ocean. We have biogenic minerals, calcium carbonate, biogenic silica. That really dominates the mass of particles in the upper ocean. And then what I'm interested in, or what I end up looking at, are lithogenic particles that may contain iron or manganese. So here shown in blue, authogenic particles, that just means formed in situ. So precipitated in situ, and these typically are the manganese and the iron oxyhydroxides that I'm interested in. But then we can also have scavenged elements that could be, I have in this orange, that could be just a coating on the outside. And so we're going to have all of these particles on one filter, and it's because we're filtering so much, they're all on top of each other. We don't have individual particles separated. And so we'll be using hard X-ray techniques in order to probe these. This is one more slide to explain basic particle dynamics to this audience, which is we may have some dissolved trace elements, manganese, iron, let's say. It could adsorb onto the surface of particles. It could precipitate and become a particle. It could be taken up by biology and become part of the biogenic material. So that's how you get from dissolved to particulate. You can also go from particulate back to dissolved by being respired. That's what remineralization means. You could be desorbed, you could dissolve. And then I mentioned that I have the small particle size and large particle size. And in my second case study, I'll talk about some differences in small and large and how that was a clue for understanding what was going on. But you have these small particles that can be aggregated into large particles that will then have this sort of composite composition of all of these individual parts. And then this is what sinks. So our sampling, analysis, and strategy is we collect these particles by in situ filtration. That results in relatively high particle loading, which helps to overcome low concentrations for XAS, but it's still hard. For the manganese bit that I'll talk about later, we had to layer as many pieces of filter on top of each other, and still we couldn't get very good data. But in general, the in situ filtration allows us to overcome some of our low concentration issues. And it gives us a lot of samples left to make other supporting measurements. And having the size fractionation allows us to understand some of the dynamics of what's going on with the particles. And because right now, well, I'm starting to get into soft X-rays, but right now, most of my career, I've been focused on hard X-rays. And the great thing about hard X-rays is, of course, they penetrate, the samples are mostly carbon, so it's a light element, and the filters are carbon or silica based, so also light elements. So hard X-rays penetrate this. So minimum sample prep is needed. And I just, for the hard X-ray stuff, just put the particles on the filter and then radiate them directly. And I don't do any further prep, which is nice. Okay, so I'm going to give you 2 case studies. One is one about iron, And I'm going to compare the iron speciation of these two regions, one in the Pacific, about 600 kilometers off the Kamchatka Peninsula over here, and one off the coast of Mauritania in the Atlantic. So that's for iron. I'm going to talk about iron speciation. And then part two, I will move to the South Pacific to this hydrothermal plume. The East Pacific rise is here, and we followed a hydrothermal plume going west. And I'll talk about manganese mineralogy. But I will pause now for questions.
Excellent timing. Yang Ha, you have a question?
Yeah, I have a general question. So you mentioned manganese usually cycles among manganese 2, 3, and 4. So I believe there's another common oxidation state for manganese, which is manganese 7. So you can have a permanganate. So my question is this like permanganate kind of like isolated from the redox cycle you mentioned? So the reason I'm asking is in the battery community, there is a like debate whether manganese can go beyond the manganese 4 through the redox cycling.
That's interesting. As far as I know, permanganate has never been found naturally. We use it in some of our geochemical extractions because it's such an efficient oxidizer. But as far as I know, we've never found manganese 7, anything beyond manganese 4. I think I saw Sam Webb at some point, and I don't know if he knows, but as far as I know, we haven't found it.
Okay, thank you.
All right, Kevin Russo, you had a question. Rosso, you had a question.
Yeah, Phoebe, I was just wondering if you know PO2 at the depth where you're sampling your iron and manganese particles.
Yeah, PO2 meaning oxygen?
Yep.
Yeah, we do. We have all of that information. And I was going to talk about a redox cycling story that we did in the oxygen deficient zone where there was no oxygen, but I ended up leaving that out. So everything I'm talking about today will be in high oxygen.
Okay.
Yeah.
Yeah, great, thanks.
But it's absolutely an important parameter that we measure regularly. Yeah.
All right, and a last question from me. You have this tens to hundreds of nanograms per square centimeter of sample.
Mm-hmm.
And I was trying to convert that into monolayers, and I didn't quite get there. Should I be thinking that a sub-monolayer, 10 monolayer?
Oh, probably more than a monolayer. I haven't done that calculation.
Okay, all right.
But it's
but it's also not pure iron or pure manganese. There's a question of what fraction of the minerals captured are actually iron bearing or manganese bearing.
That's right. And in fact, most of it is not. So we have most of our particle mass is not the iron or manganese.
Hence the need for microfocus.
That's right.
Okay, thank you.
Mm-hmm.
You should continue. It's a great start. Thank you.
Sounds great. Okay, so I'm gonna start with part 1. And this is pretty old now. It's over 10 years old, and and synchrotrons have beamlines have progressed since then. So they're more capable, but I'll sort of take you back to my brain 10 or 15 years ago and walk you through how I was trying to get my head around how to characterize the speciation of iron everywhere in the ocean with limited beam time. How do you do that? Okay, so I'm going to compare these two places, one in the Pacific, one in the Atlantic. And let's start in the Pacific here. And so standard oceanographic thing to do is we plot concentration profiles, and because we are thinking depth, we have depth on the y-axis and our concentration on the x-axis. So that always trips my students up. So think going down. So here's the surface of the ocean, and we have high concentrations at around 185 meters in this particular location off of in the Pacific, Northwest Pacific. So as an oceanographer, we want to understand why is there a concentration maximum here? And if we study this concentration maximum, will we understand something about how it got there? And is this, for example, the iron here the same as the iron here, just at higher concentrations, or is it something different? And if it's something different, is it going to tell us something about where it came from? So here's an XRF map of a sample from this sample. You see lots of micron scale hotspots. Each pixel is maybe, I don't know, a few microns, probably 5 microns or so, a few microns. And you see very abundant hotspots. So lots of iron to look at. How do we start? So you can focus, you can shine a bulk XAS beam, and then you'd get the average speciation. And I would say that that's probably the best way to just go through and characterize how is the speciation different as you go from surface to deep, because it's fast. You get average speciation, you get a broad view understanding of how things are changing with depth. But then, of course, if you do a bulk XAS, then you miss all of the detail, and you don't know whether this dot is the same as that dot, for example, and maybe that's important, maybe it's not, but you lose that information. So, okay, so if you want to know whether those two dots are the same, you can do zanes on those individual dots, and that's great. Now you know that this dot is different than that dot, but what about all the other dots? You know, what is the most representative dot to look at? And so that's why 10 years ago, when I learned about chemical species mapping, I was so excited. So now we're doing XRF mapping, but we're doing it at slightly different energies to take advantage of the fact that different iron species have slightly different edges and different features. And so now we're trying to pull out what those features are and get a lot of statistics. So instead of doing individual zanes on individual spots, now we're just doing mapping at different energies in order to get these different spots. And I just learned that now you can, at SSRL, you can get a small little zanes at every single spot. Oh, that's crazy. In any case, at the time, we were doing just a few energies, and that was enough to pull out these three broad groups of iron. So anything in red, which you don't see, oh no, there's one. There's a tiny little red spot right here, which is pyrite. And anything in green is an iron 2, and anything in blue is an iron 3. And so you see a fairly good mix of iron 2 and iron 3. These two spots happen to be different. This one's 2, and this one's 3. And you see that how it's distributed. You can quantify this and get an average oxidation state, add up all the counts in your iron 2 channel, all the counts in your iron 3 channel, and then look at how that, what the average is. And it actually compares pretty well with a bulk. And so we did that. And so if you go back to this concentration profile, we did both the chemical species mapping and bulk XAS on most of these samples down this profile to try to understand whether it was changing or not. And that's what this is. And we're showing you, I'm showing you the percent iron 3, 100% is all iron 3. So this is 60% iron 3, 40% iron 2. And you see that where there was that concentration profile maximum, we have this minimum in percent iron 3. The dark dots are showing you the average oxidation state, or the percent iron 3 from the chemical species map. The red are two different ways of interpreting the bulk XAS data. There is some slight variability, but they're telling you the same story. So I'm quite encouraged by that. And I now in my current life, we mix and match the two depending on what beam time we have available, what sample type we have. We will mix and match with some caveats. We spent a lot of time making sure that the answer we get from one technique is comparable to the answer we get from another technique and from another beamline and from another synchrotron. This worked. This was data from SSRL.
A quick interjection. Just so you know, we can't see your cursor.
Oh, you can't?
No, I've understood what you meant when you were talking about specific. There you go. Now I can see it. There we go. Thank you.
Okay, thanks for letting me know. Okay, so here we go. Thank you. So black is the chemical species, red is the bulk. And so we spent a lot of time making sure that this number is comparable to that number. Okay, so this told us that we had something funny going on that was special about this 185 meter concentration max. This is what the chemical species maps look like. Now we wanted to dig in and dive in and figure out what it was. What is that iron 2 stuff that's going in? Is it some sort of internal redox cycling, or is it something transported from somewhere else? And so we did 209 zanes, which took several beam times. And so each of these represents some sort of a zanes spectrum. And I was overwhelmed by how to get my brain around it. And so I worked with Matthew Marcus, and he developed this sort of clustering visualization analysis that allowed us to see the relationship of all of these different zanes spectra. And so we did this clustering, and we're able to reduce those 209 spectra to 4 different clusters. And then we can fit those clusters and see that these were the main groups that were showing up. We had iron 2 phyllosilicates, we had mixed iron 2 and 3 minerals, we had iron 3 clays, and we had these mixed clays, oxyhydroxides. What we didn't have really were a lot of indications of any sort of iron 2 oxides that might have participated in some sort of in situ redox cycling, nor did we have a much indication of some sort of biologically bound iron 2. What we saw were crystalline iron 2 minerals. So that told us, so now putting it together, here's that concentration profile, here's that average did you lose my laser pointer again? Do you see it now? No?
No.
Gosh, why is that?
I don't know. The modern world is annoying sometimes.
The modern world is annoying. It seems like I might
now
have
it's back.
Now I have to turn it on every time I switch screens. That's annoying. Okay, so here we have that minimum in the average oxidation state, that maximum in the concentration. And what we interpreted that to be was, if this is a sort of schematic cross-section of the continental margin, here's the shelf, here would be the surface ocean. We thought that since this is the location of our sample, right off of this is the Pacific Rim of Fire, this volcanic margin. And volcanic material has a lot of crystalline reduced minerals. And we took this to mean that there was a transport of this volcanic material from the margin into the interior of the ocean. So this allowed us to understand, at least in this location, and of course, at least in this location, let me turn on that laser pointer again. In this location, in the Pacific, we're surrounded by this volcanic margin, and we saw evidence for this transport from the margin into the interior of the ocean. So I wanted to go to a very different margin, and this is now off the coast of Africa. This is not volcanic. It's very weathered, very old, lots of iron 3 material, for example, here off the coast of Africa. And just a sort of reality check. What do we see there? And here, just to remind you, on the left is the Pacific profile that I just showed you. Again, with in the black is the total particulate iron, and in the dark blue is the iron 3. Is that right? Yes. In the green is the iron 2, and red is pyrite. So you see a mix, pretty much a 50-50 mix of iron 2 and iron 3. And then now if we go off the coast of Africa, you see that just visually it's very obvious to tell it's all blue. So it's all iron 3. Now here's black is the total iron, and blue is showing you how much of that is iron 3. So we have extremely different speciation in these two margins. And what I took that to mean is margins are important. They do supply iron to the interior. Depending on the lithology of that margin, you're going to influence the iron that is in the ocean, and have the particulate iron that is in the ocean will reflect the iron that's coming from the margin and its mineralogy. So the summary here, depth variations showed that there was depth variation in the speciation and the mineralogy. It showed us that volcanic sediments from the continental margin in the Pacific are an important source of iron to the interior. We saw lots of heterogeneity in iron speciation. I would say most of the iron is in iron 3 form unless you're very close to some sort of volcanic sort of input. And sort of going back to that beginning bit of thinking about iron and its bioavailability and its role in productivity, the mineralogy of particulate iron is important for bioavailability. Particulate iron is the biggest pool of iron in the ocean. The dissolve phase is quite low. So until now, most models don't consider it as a potential source of iron as a micronutrient. There was a recent paper that showed that, in fact, particulate iron 2 is more bioavailable to a diatom, a phytoplankton, than say an iron 3. So understanding that distribution of mineralogy, where in the ocean, whether it's in a place that is iron limited, can have implications for how we think about modeling it, for the role of iron for controlling productivity. So I'm going to pause again.
We have a question from Matthew Marcus.
Okay.
Okay, you mentioned crystalline iron too as being produced by volcanoes, but what about glass? After all, glass is more soluble than crystal, so I would think that would be pretty important.
Yeah, so I don't ever see something like, or almost never see anything like olivine or glass. And I take that to mean that it's so soluble that it's gone away. By the time I get it, it's not there. So what I end up seeing is the residual stuff that has been weathered or dissolved after the extremely available and soluble stuff has gone away.
Right, so it can be an important source, but you just don't see it because it's already used, it's already dissolved.
Yeah.
James Evans, you have a question?
Yeah, I may have missed this, and I'm sorry if I did. So why would the volcanogenic materials from the Northwest Pacific accumulate in that 200 to 300 meter depth zone?
Good question. I think that has to do with the circulation. And so you have something about the circulation and the resuspension of shelf material allowed it to be efficiently transported. Water tends to move along constant density surfaces, and density in the ocean tends to increase with depth. And so water moves horizontally, and something about the resuspension of material at that particular density surface and the availability of currents at that density surfaces was able to transport that efficiently into the interior. So you do need the right circulation for that to work.
Okay, not seeing other questions. You should continue.
Great. Okay, so part 2 is moving on to manganese, and this was work led by my postdoc, Dr. Zhongmi Li. And so now we're going to the South Pacific. The East Pacific rise sort of runs around, you probably don't see my cursor. Okay, let's try that again. The East Pacific rise runs here. The cruise that I'm talking about was in 2013, and it crossed this mid-ocean ridge and then followed a plume going westward. The background concentration colors here are showing you helium, which comes from, there's a mantle source of helium. So it's a very good tracer for hydrothermal influence. And this is now a cross section, sort of like the cross sections I showed before, but now instead of north-south, we've got east-west. This is east starting here, west going over here, depth again on the y-axis, and the colors are showing you the concentrations. And this is helium. So this is showing you that there was a hydrothermal plume and that plume traveled westward. This paper made it into Nature because of the iron, and I didn't actually show the iron, but the iron also showed this incredible many thousands of kilometer transport. And no one expected that because, as I mentioned before, I said iron is very insoluble. It should oxidize. It should be taken out of seawater. And maybe you've seen some talks from Brandy Toner who may have talked about that. I'm going to focus on the manganese. So just as a review, and as someone asked, so there's 3 main states of oxidation states of manganese that we find in the environment. Others exist, but these are the ones we find. Iron 2 is soluble. It gets oxidized to iron 3, which is unstable, which gets either reduced back to 2 or oxidized further to manganese 4. Did I say iron? I meant manganese. It's thermodynamically favorable, but kinetically inhibited. Actually, I think one of these steps is not thermodynamically favorable. So the entire thing is thermodynamically favorable, but you require catalysis in order for that oxidation to happen, usually microbial. So even though manganese 4 is the stable form, you end up finding quite a lot of manganese 2 around just because that oxidation is very slow. Once you get to manganese 4, it's quite insoluble and will precipitate as a manganese oxide. And as I mentioned before, manganese oxides have extremely high specific surface areas, and they can act as very efficient adsorbents for lots of trace metals. So what we wanted to do was to go, so here's now that helium section across the, sorry, let me make that laser pointer. Okay, here's that helium section showing you where the plume is. Here's showing you the dissolved manganese. So there is certainly a very high source of dissolved manganese. And then here's our data showing particulate manganese. It's a little ugly, but this is saturated, but you see a very, very high particulate manganese bullseye right here. But you still see some particulate manganese that's also being advected west in this plume. So we wanted to use this as a natural laboratory to study trace metal scavenging by manganese oxides. We know it's coming out in a dissolved form. We know it's getting oxidized, and then it's being transported. What can we learn about how oxidation affects scavenging by looking at manganese oxide and what elements it's associated with along this plume? So I'll be talking about samples from the near field, so right above the hydrothermal vent, well, within some tens of kilometers from the vent itself, and then the far field plume. Most people think about the far field as being out here, but because of the limitations of our concentrations, this is what we call the far field. And in fact, we show that the mineralogy is really quite different even by here. So far field is everything that's basically farther than 100 kilometers, and that starts here. So I'll have this kind of, for the next few slides, this kind of color coding. The near field will be on the right, just because in the space we've got, the near field is further right, further east than the far field. So we'll have the near field data on the right and then the far field data on the left. The near field data, what I'm showing you here is particulate, this is just concentrations now, no speciation, just concentrations that we measure using a mass spec, dissolution in mass spec. On the x-axis is particulate manganese concentrations, on the y-axis is particulate cobalt concentrations. Manganese and cobalt have been found to be very well correlated in the ocean. And indeed, we find this fantastic correlation between manganese and cobalt in the near field. And then we go into the far field and the correlation just drops away. The triangles are the small particles, and then the squares are the large particles. So in the near field, you saw a correlation in both the small and the large particles. In the far field, even though this looks like it might form a line, it's actually, if you look within the small particles and you look within the large particles, there is no correlation at all. So our association just drops off completely. You find the same thing for molybdenum. It looks slightly different. And in fact, the association of molybdenum to manganese is quite different as a mechanism as association of cobalt to manganese. But you still find this fantastic correlation in the near field. And then, whoops, wrong direction. And then the correlation just completely breaks down in the far field. So there's something different about the manganese in the near field that is allowing for this close association with these other trace elements. And we see it for other elements too, protactinium, thorium, but lead, but I'm not talking about that. I'm just using these two as examples. Another thing we looked at, and I mentioned the size partitioning, is we looked at how much manganese, again, this is just concentrations, no speciation yet. These are now concentration profiles. Let me get my cursor back. Concentration profiles, so depth on the y-axis, concentration of manganese on the x-axis, and the near field goes from 0 to 5. These are in nanomole per liter, and then the far field goes from 0 to 0.5. So really the concentrations drop off by a factor of 10. And then these two symbols are showing you large particles, so greater than 51 micron, and small particles less than 51 micron. And you see in the near field that the small and the large are about the same. That means 50% of all the manganese is in large particles, 50 in small particles. If you go into the far field, you see that it's dominated by the red, which is small particles. We have most of the manganese in the small particles, not in the large. So this is an extremely unusual size partitioning. This is much more typical of what we find in terms of just bulk mass. Particle mass is usually about 80% small, 20% large. And so the manganese was doing what particles normally do. But in the near field, it was really quite different. Okay, that's that. Just as a summary, in the near field, we have this abnormally large size partitioning of the manganese. We had the strong correlation. In the far field, we had normal size partitioning for the manganese and no correlation. So what we wanted to do is we clearly have these different phenotypes. We wanted to understand whether looking at the mineralogy could help us understand whether there was something that was different about the manganese oxide and whether we can understand on a process level what was going on to make these two different parts of the hydrothermal plume different. So here, I think we were using bulk XAS at SSRL in order to look at the basic manganese oxidation states. And actually, if you just calculate the average oxidation state, so blue is the proportion, so the, let me explain. So the y-axis is depth, the x-axis is concentration for, let me get the pointer back. The top axis is concentration of particulate manganese, and you should read the circle symbols. So that shows you this plume, this manganese plume right in the center, concentration max right in the center of the plume. And then we, for every one of these bars, that's a different sample, we looked at the distribution of that particulate manganese in the manganese 2, 3, and 4 form. And that's plotted as these stacked bars. So that goes from 0 to 1. It adds up to 100%. And this is just showing you the fraction that is in manganese 4, 3, and 2. So that if you were just to calculate an average oxidation state, it's actually fairly similar between near field and far field. But if you then partition it between 2, 3, and 4, you see that the near field has a lot of manganese 3, and the far field has very little manganese 3. So that was a clue. That was interesting. We didn't know what to do with that, about that. And then in the near field, we had, let's see, 4 nanomolar of 3 to 4 nanomolar of particulate manganese. That's enough to do XAS. It's painful. It takes all day, but it was enough to do XAS. So we based our interpretations on this work by Lehrman et al. In 2011. This is not our work, in which they grew bacterially, they precipitated manganese oxide in the lab using bacteria, so it was bacterially mediated. And they looked that, they brought this to the synchrotron, and then they looked at the mineralogy of that bacterially precipitated manganese oxide. They found it was highly reactive. It had this hexagonal symmetry and is mostly in the manganese 4 form. But then, so they formed this, I guess they formed it at the synchrotron, and then they exposed it to light, and then they put it back into the dark. Sorry, let me get this pointer back. So they exposed it, when they first formed it, you see this characteristic hump at around a K of 8. And then as they incubated this product in the light and then in the dark, they saw this hump go away. And so they saw that it ripened to be a less reactive form, and it gained triclinic symmetry, and it had a much higher proportion of manganese 3. And so that was our clue, is that we had this place where we had manganese 3 in the near field and in the far field did not. And in fact, when we did XAS on our own sample, so this is now a sample from one of our near field stations right in the middle of that plume. The blue is a standard hexagonal bernicite, manganese 4 bernicite, and you see that hump at a K equals 8. The red is another standard, which is showing you triclinic bernicite, and then this is our actual sample in the black, and you see that we don't have that hump. So we also seem to have this ripened triclinic manganese oxide that had manganese 3 in it. And the reason we were excited about that is because the way that the ripening happens is that you form this manganese 4, get the pointer back. You form this nope, pointer's not back. You form the manganese 4, it's very reactive. Manganese 2, which is dissolved, will adsorb to the surface and then react with that manganese 4 and become manganese 3. Cobalt can compete with manganese for that same surface site and react with manganese 4 and become cobalt 3. And so this is now a mechanism by which the mechanism that causes the manganese 3 to be incorporated into the mineral, the manganese oxide mineral, is also the mechanism that incorporates cobalt into the mineral. And this is work by Tanaka et al. Showing that this happened, is they added cobalt to a fresh delta MnO2 bernicite. They showed that at first shows up as cobalt 2 that's adsorbed to the surface, and then over time, 48 hours later, it becomes cobalt 3 and gets incorporated into the mineral itself. So this seems to be happening to us then in the near field. We have abundant manganese 3 that shows that we had this really fresh bacterially produced manganese 4 oxide that then ripened and incorporated more of this manganese 3 in. Meanwhile, cobalt competed with that manganese to be also co-oxidized and incorporated into that manganese oxide. And that then it explains that very tight correlation between manganese and cobalt in the near field. The thing that size partitioning comes in here because this was so reactive that the manganese oxides grew very, very quickly. And once you get big, then you sink like a rock. You become a big rock and you sink out. And so you can't be transported out west. And so what we think happens in the west, in the further part of the plume, is that the non-reactive, whatever non-reactive portion is left over from the near field is not growing. It's not ripening. It's not growing because it's not reactive. And as a result, it stays small and it can be transported. It doesn't sink out. And it wasn't reactive to begin with, which is why it didn't grow. And so it's being transported out west. And now we have this sort of residual non-reactive stuff that is out west and is not scavenging. It's no longer actively incorporating other trace metals such as cobalt or molybdenum. So the summary here is we've got fresh biogenic manganese oxides that form. We found that they form in the near field plume. They're extremely reactive. They can grow very quickly and as a result sink out, so they don't get transported very far away, but then they can co-oxidize cobalt and adsorb molybdenum. In the far field, we have this much less reactive stuff that is, we would have expected it to, because I guess as an oceanographer, I always think of manganese oxides as reactive everywhere, as being these sponges that are always pulling trace elements out of solution. But what we're finding here is it's really only the fresh stuff that is super reactive, the sort of old unreactive stuff. There's still unreactive stuff that's out there that may not be as efficient as we think it might be to pulling out other trace metals. So stepping back then, synchrotron XAS techniques, they're ideal for probing the redox speciation and mineralogy of iron and manganese in the ocean. I just gave you 2 little vignettes, but knowing the mineralogy and redox state of iron in the ocean is helping us understand whether this pool is bioavailable, how it should be incorporated into climate models. And then for the manganese bit, this is now on the other end is not the source to biology, but sort of the removal of trace elements. We found that freshly precipitated manganese oxides are very efficient scavengers, but old manganese oxides are not. And that's it for me.
A great talk. Thank you. We'll start with a question from Matthew Marcus, and I'll remind everyone, please type your questions or small proxies for your questions into the chat, and then I'll ask you to unmute. Matthew, please.
Yeah, so there seems to be lots of manganese too. Where does that live? Is it bound to organics like iron 2 can be? Or
Yeah.
Okay.
We have a mystery. We also have a mystery iron 2 species that is not organic, and we tried our darndest to figure out what it was. The concentrate, we tried to do XAFs on it. And actually, we did do XAFs on it, and it did not look like anything that we could find in any library that we could scour. So we do have a mystery manganese 2 bearing mineral, and we have no idea what it is. Some of it in the far field is absolutely organic, but the near field stuff is too high in concentration. It's not organic, and we have no idea what it is.
And it's not manganese 2 sorbed to manganese oxides?
No, because we had that as a standard, and we made all sorts of sorption standards, and it didn't look like any of our sorption standards.
Cool. A mystery.
Yeah.
Okay, looking for more questions. I'll ask one. On several of the plots, you showed how the helium-3 concentration varied with position. Can you just tell me a little bit about why that's the important marker and where the helium-3 comes from?
Yeah, there's a mantle source. It's primordial, and so it's from the creation of the Earth, and it's leaking up, it's escaping from the mantle. And it's just, the great thing about helium is that it's a noble gas, so it's very unreactive. And this particular one, well, it has it has a source in the atmosphere from the decay of tritium from bomb, but deep in the ocean, the main source is from the mantle, which means that basically the only source is hydrothermal plumes. And so it's an excellent tracer of any hydrothermal input. And because it's a noble gas, it's completely unreactive. So its decrease in concentration here is simply due to dilution. So it's a tracer of where the water is going. And as the concentration is decreasing, it's being diluted. And so we use that as what we call a tracer of conservative behavior. And you can plot the concentrations of manganese versus helium. And if it's a straight line, then we know that manganese is just being diluted just like helium is.
I understand. I understand. Thank you. That's terrific. That's terrific. All right. We are at 10 a.m. And so let's thank Phoebe again. It was a terrific seminar. Thank you very much.
Thanks for having me.
And I will stop the recording